So my previous postdoc, Ken Harris, did a very beautiful article, already calculated this, that, for example, in the auditory cortex, the realm of possibilities, that is what patterns exist, is not infinite. It depends on the brain, and you can look at it during sleep, for example, because sleep is the honest expression of the brain. It doesn't lie, like we can do when we are conscious. And then it said every single pattern that we observe when you evoke firing in the neurons with any frequency, any auditory pattern, falls within the range of these possibilities. Mm-hmm. And then another student, and there was a student of mine who was at about the same time when we were in the lab when we were discussing the same kind of thing, George Dragoy. He went on and he looked at the hippocampus view, and he also showed that indeed there are patterns in the brain that can predict what happens to those neurons in a novel environment. And then this is the theme that we are working on ever since, that indeed, if you look at the brain this way, it's more economic and more easier to explain. And inhibition comes forward as a organizing force. And plasticity, as a very important thing, is not abused that much as it used to be.
Let's talk a little bit about inhibition rhythms and things like sleep and memory. So one of the ways I want to steer you here is you've done a lot of work on brain rhythms, a lot of work on the different phases of waking and sleep. And one of the things, we'll just sort of assume people are familiar with things like EEG here, and that obviously when you record EEG activity, you can record from large ensembles of neurons rather than individual neurons. And you can do something like EEG. And when you do that, when an animal's awake, when it's sleeping, when it's in non-REM sleep, when it's in REM sleep, you see different combinations of rhythms that define each of these brain states or behavioral states. How do you start to think about the relationship between those brain states, the rhythms they contain, and what those are allowing or disallowing when it comes to memory consolidation and recall?
What you said is absolutely right. You can find all sorts of rhythms. And again, history here is very interesting, because when people were talking about rhythms, they involved mostly people who looked at very simple organisms, which have a lot of rhythmicity, such as the gastric ganglia or locomotion. People who were obsessed about the human cognitive abilities, they looked at the EEG and they said everything that happens when you are doing something useful, which is cognition, gives you a flat EEG. Rhythms occur only when you are asleep or when you are epileptic. So that was the state of the art until 1970s. And when you use higher resolution electrodes and better designs and so on, and again, hippocampus is an interesting thing, because if you stick an electrode into the hippocampus, you cannot not see this huge theta activity. It was actually discovered, at least in a freely moving animal, by my mentor, who was the first one who looked at how theta could be related to any behavior.
So theta activity, that's a particular brain rhythm that you see prominently in the hippocampus.
Yes, exactly. And that was a great lesson to me, because my mentor was an extraordinary man, Andrei Grosjean, who looked at it and said, oh, this is related to the orienting response of the animal, which is a Pavlovian term, and it's a response. And there was another person at the other end of the world in Canada who became my postdoc mentor, who said the opposite. No, it says it's related to the voluntary action. And so one was the perception side, one was the...
Yeah. So again, it's this clash between something being evoked from the outside versus something being present from the inside already.
Exactly. So that was good thing.